chapter, we are going to see in details the male reproductive system. So the male reproductive system is composed from three elements: the testes, which are the male gonads, the male genital tract, and the accessory glands. Okay, so we have the testes, the male genital tract, and the accessory glands. The testes, they produce the spermatozoa, as we explained before, and okay, it is written like this in the plural, and in the singular it is is. Okay, they produce spermatozoa, and they contain the Leydig cells secreting the testosterone. As we said, they contain the seminiferous tubules, which inside of them there is the Sertoli cell. There are the Sertoli cells and the germ cells also, but also they contain the Leydig cells, which secrete the testosterone. And we have also the male genital tract, which ensures the maturation of the spermatozoa. The maturation of the spermatozoa takes place in the epididymis, as we are going to see in this chapter, and the genital tract ensures the transport of the sperm cells of the spermatozoa from the testis to the penis, okay, for their ejaculation. So they ensure their transport through the vas deferens, the ejaculatory ducts, and the urethra. Okay, and then we have the accessory glands like the seminal vesicle, the prostate glands, and the prostate gland and the Cooper glands, which secretes the seminal fluid. Okay, the seminal fluid contains nutritive elements for the sperm cells. Okay, and this seminal fluid will be mixed with sperm cells to form the semen. Okay, so the accessory glands, they produce the seminal fluid, and we will see each one. We will talk about each one of them later. So here we can see the male reproductive system. We have the testes, they are here, okay, and then so the testes, they are the first element. Then the genital tract composed from the epididymis. You know, the epididymis is directly on the upper side of the testis. Here is the epididymis. Then we have the vas deferens here. This channel, this tube is the vas deferens, in which the spermatozoa will pass through it. Okay, and then we have the ejaculatory duct. The ejaculatory duct هو is the يعني هو بكمل vas deferens, but after the urinary bladder. يعني بعدين بصير في عنا the two vas deferens will diverse in the ejaculatory duct here. Okay, and then we have the urethra. Okay, the urethra اللي هو the ejaculatory duct will كأنه they will fuse, okay, in one duct called the urethra, and the urethra opens at the end of the penis. Okay, and the urethra is transport the urine and the sperm cells. يعني both of them, they pass through the urethra in the men. In the females, it's different. It's only for the urine. And now the last element is of the male reproductive system. We said they are the accessory glands, which are the prostate gland. Here we have the prostate gland, the seminal vesicule. We have two seminal vesicles, and one on each side, and the Cooper's gland, or which is also called the bulbourethral gland. We will see it later. And also this bulbourethral gland will secrete fluid that will, in fact, will pass through the urethra to clean it, to eliminate the acidity of the urine. So the sperm cells or the semen can pass without hurting or without damaging the sperm cells. Okay. Now we are going to see in more details the organization of the and the structure of the testis. This is one testis. Okay, it is the testes are ellipsoid in shape. They have an ellipsoid shape. They are located in the scrotum. You know, this pouch outside of the body, this pouch of skin is called the scrotum, and the testes are localized inside of the scrotum, outside of the body, and this localization is important for the spermatogenesis because, as we said, if you remember in the last chapter, then in the scrotum the temperature will be 2 to 4 degrees lower than that of the body, and this lower temperature is essential for the good occurrence of the spermatogenesis. If the testes remain in the abdominal cavity, were to remain in the abdominal cavity due to the a little bit higher temperature in the abdominal cavity, the we will have problem at the level of the spermatogenesis and in the sperm cells, and the person will be infertile. So this position in the scrotum is important because the temperature is 2 to 4 degrees lower than that of the body. Okay, and then on the upper side, on the posterior superior region of the testis, so on the upper side and posterior, on the posterior upper side of the testis, we have the epididymis. Okay. Which is a channel also in which the maturation of the sperm cells will occur. We have the epididymis positioned here on the posterior superior region of each testis, and then we have the testis is covered by this fibrous capsule. So this is inside of the testis. If we do a cross section, we are going to see the seminiferous tubules here, if you can see them. Okay, and if you look from this side, here you have a cross section and here you have a longitudinal section, and from this side here there is no ما في ولا section. Okay, ما في section هون. This is the testis, how it looks from outside. It is covered from a fibrous capsule or from a fibrous element called the tunica albuginea. Okay, so it is covered by this tunica albuginea. The tunica albuginea is composed from collagen fibers and contains some smooth muscle cells. It contains a lot of fibers, collagen fibers, and smooth muscle cells. Those smooth muscle cells will allow the contraction of this tunica albuginea, and then will help in the ejaculation of the sperm cells from the seminiferous tubules toward the epididymis. Okay, so the smooth muscles in the tunica albuginea are important for the propulsion of the sperm cells from the seminiferous tubules into the epididymis. And this tunica albuginea is also innervated by adrenergic terminations. So it's innervated by nerves, and the nerves of those, the termination of those nerves are responsive to adrenaline and noradrenaline, and that's why we call them adrenergic terminations. Okay, so this tunica albuginea is innervated by adrenergic terminations, and it contracts spontaneously and rhythmically, helping to propel sperm and helping to propel the sperm and the testicular fluid out of the testis. So it can contract to propel the like we said, the sperm cells and the testicular fluid, because there is a testicular fluid also in the lumen of those seminiferous tubules, which will be propelled with the sperm cells to the epididymis. Okay, so this tunica albigenia contracts rhythmically and spontaneously to propel those spermatozoa with the testicular fluid. Now we have all. Yes.
بس إذا ممكن تدلينا على السكروتوم بالصورة قبل ما تقلتي السلايد الثانية.
هون ما في مش راسمين السكروتوم، هون بس في testicle with the epididymis and the vas deferens. هون the blood vessels reaching the testicle. But the scrotum هو هون يعني من برا، هو outside، هو skin من برا. اوكي، testicle جوا بقلبه. نحن كنا we were observing the testicle. The scrotum هو is the skin that covers يعني where the testicles sit. اوكي، وفي عنا two testicles بيكونوا two testicles next to each other. اوكي.
اوكي دكتور، yes تمام.
In this figure we don't have the scrotum or the scrotal pouch. It's not represented. And then we have the Highmore body. What is the Highmore body? It is a thickening of this albigenia, of this tunica albigenia. This tunica albigenia will get thickened here on the upper posterior side, okay, just between the seminiferous tubules and the epididymis. In this region here, it will get thickened, okay, and this thickening is called the Highmore body, okay, the thickening of the tunica albigenia. And this Highmore body is perforated by channels coming from the seminiferous tubules. As you can see, the seminiferous tubules will extend to give, here we have the straight tubes, okay? So the, let me show you where. You have the straight tubes coming out from the straight tubes here, okay? So the seminiferous tubules will converge to give the straight tubes that will reach the Highmore body, form this channel canal in the Highmore body, okay? So those channels in the Highmore body, plus we have also blood vessels that reach this Highmore body here, they constitute together the reed testis, okay? So you have here this figure. Here we have the testicle, and this is the epididymis, okay? And this is the upper posterior side of the testicle. On this upper posterior side, all around the testicle we have the tunica albigenia. Here it is covering it also, but imagine here that you have a longitudinal cross section, so we are seeing what's inside. Okay, هي tunica albigenia من برا كمان، بس هون عم نشوف بس يلي جوا، and we are seeing the edges of this tunica albigenia. In yellow, at this region here, this tunica albigenia is thickened. In yellow here, it will form the Highmore body, okay? And those seminiferous tubules will extend by those straight tubes or straight tubules to reach these Highmore bodies and forming like a web of channels here inside of this Highmore body. This web of channels is called the reed testis. And also the reed testis contains blood vessels, okay? So we have channels conducting the sperm cells and also blood vessels forming this reed testis. Okay, فهمنا شو الهايمور body and what are the reed testis?
دكتور، يعني نحن فعليا نشوف عملات
سوري، ما الصوت قطش، ما فهمت عليكي.
فعليا ما بنشوف انه في هايمور بودي من نرمز له بالريد تستس.
لا, there is a Highmore body, but the reed testis are inside of the Highmore bodies. يعني هن مثل a network of channels inside of the Highmore body.
يعني هو الهايمور بودي هو بس thickening منه إنه actual شيء يعني.
Yes,
إنه
it's only a thickening of the tunica albuginea, and in this thickened region of the tunica albuginea, we have this network of channels and blood vessels forming the reed testis. We call them the reed testis.
اوكي دكتور ماشي.
Okay.
Thank you.
دكتور بس شغلة حكيتي شي عن السمنفرس تيبيولز وين موجودين يعني؟
هولي كل هولي يلي هون in blue أو in شوي purple هون they are in pink. Okay, those are the seminiferous tubules.
يعني هن canals ولا شو؟
Yes, they are channels. هن we said before in the chapter 1 and in the last chapter يعني in the chapter 3 that the spermatogenesis will occur inside of those tubules. يعني the sperm cells will be formed inside of those tubules, and all the germ cell lines, all the cells of the germ line are inside of those seminiferous tubules. يعني كل شيء spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and spermatozoa are inside of those tubes and not outside. Okay, and the Sertoli cells also are inside of those tubes, but the Leydig cells will be outside of the tubes between the different between the different ما بطلت أعرف حاجة. between the different seminiferous tubules. Okay.
Yes, doctor, clear. تمام مش الحال.
Okay. And then we said we have the tunica albuginea forming here thickening on this on this upper posterior side, and also this tunica albuginea will extend to the inside of the testicle between the seminiferous tubules. يعني it will go inside between those seminiferous tubules. As you can see here, the seminiferous tubules are not filling all the inside of the testicle. يعني they are organized. شوفوا كيف الorganization تاعيتهم. So between them we have tunica albuginea, extensions of the tunica albuginea. So in gray you can see in gray. Here in gray, okay, this is the tunica albuginea extending between the seminiferous tubules, okay, and those extensions to the inside of the tunica albuginea are called septa. They are forming septa. We say that the tunica albuginea is forming septa between the seminiferous tubules, and the singular of septa is septum, okay. So the tunica albuginea give rise to numerous incomplete collagenous septa, يعني stretched divisions. So it will, the tunica albuginea from the outside, it will extend and form incomplete collagenous septa. يعني ما هي يعني formed from collagen fibers. Okay, so we have septa or divisions that will separate the seminiferous tubules. Okay, so those septa will divide the testis into about 250 testicular lobules. And so those septa of tunica albuginea will separate this, will divide the testis into 250, almost 250 testicular lobules. Each testicular lobules is composed from 1 to 4 highly convoluted tube of seminiferous tubules. So here we have one, you can see we have one lobule, okay, one lobule of the testis. This is one lobule, this is another lobule, this is another lobule. Okay, so in each lobule we can find one seminiferous tubule to 4 seminiferous tubule, يعني 1 to 4, and the tubules are convoluted, يعني they are very convoluted tubes inside of those lobules. Okay, so those septa will divide this testicule into 250 lobules. Yani 250 groups of seminiferous tubules. Each group will contain 1 to 4 seminiferous tube. Okay, and when we say about 250 testicular lobules, we mean it can be between 200 and 300. The average is 250. And we said that the spermatozoa or the sperm cells are produced inside of those testicular seminiferous tubules. Okay, and we talked about the straight tubes, which are extensions of the seminiferous tubules. The seminiferous tubules will converge toward the hymore body, where they reach the reed testis by those straight tubes. So here we have the seminiferous tubules. They will converge to the reed testis by the way of those straight tubes. Okay. Now, the seminiferous tubules are the place where the spermatogenesis occurs. Okay, inside of those tubules, this is a cross section of a seminiferous tubule. Inside of this tubule, we can see the organization. Close to the periphery, we have all the germline cells. Okay, and all the germ cells close to the periphery and a little bit extended to the middle, but in the middle of this tube, it's a hollow tube in the middle. And since it is tube, it should be a hollow from the middle, empty from the middle. Okay, in the middle, we don't have those cells, but later when the sperm cells are formed and they get mature, they will be released in this lumen, and due to the contraction of the tubes and of the testicles, they can be transported to the epididymis. So each tube contains germ cells in close relation with Sertoli cells. As we said also that we don't have only the germ cells inside of the testicule, and germ cells, we mean the spermatogonia, the primary spermatocyte, secondary spermatocyte, the spermatids, and the spermatozoa. We have also the Sertoli cells, which are somatic cells. Okay, we have the Sertoli cells inside of the seminiferous tubule. Here you can see we have the germ cells, spermatogonia, primary spermatocyte, secondary spermatocyte, and spermatids. And we have also the Sertoli cells. The Sertoli cells are large cells. Okay, we cannot see the periphery, the edges of those cells, because they extend under the germ cells, and they are in tight contact with them. Okay, and they are very large cells, but here this is their nucleus. What we can see here is their nucleus, but everything in purple are the Sertoli cells. Okay. And as you can see, and here is the periphery of the seminiferous tubule. This is a section from the seminiferous tubule. This is the periphery of the seminiferous tubule. Here it is represented. And more the cells are, the cells which are closer to the periphery of the tubules, they are more immature. Okay, and the most stem cells, we will find them directly adjacent to the periphery of the seminiferous tubules. And the more they get mature, the more they will migrate toward the lumen of the seminiferous tubule. Okay, and the more they will get mature, we will find them closer to the lumen. Now, each seminiferous tubule is limited by a basal lamina and a lamina propria. Okay, the basal lamina is this line, and like the basement membrane. This is the basal lamina of the seminiferous tubule, and a lamina propria. The lamina propria is composed from fibroblast cells, collagen fibers produced by those fibroblast cells, and from another type of cells, which are the peritubular cells, which we call also the myoid cells. So inside of this lamina propria, we'll find collagen fibers, fibroblasts, and also peritubular cells. Okay. The peritubular cells, they are like muscle-like cells, smooth muscles-like cells. They resemble, or they are similar to smooth muscle cells. They are able to contract, so they are contractile cells. Those peritubular cells, they are contractile cells, and their contraction will help also in the contraction of the seminiferous tubule and in the release of the sperm cell, or the propulsion of those sperm cells produced into from the seminiferous tubule into the epididymis. Okay, so the peritubular cells, they are contractile and participate in the evacuation of spermatozoa from seminiferous tubules to the reed testis and then to the epididymis. Okay. So here we have the inside of the seminiferous tubule, composed from the germ cells and from the Sertoli cells. The periphery of the seminiferous tubule is composed from a basal lamina and from a lamina propria. The lamina propria contains fibroblast cells, peritubular cells, and collagen fibers. And now, between the tubes, between those seminiferous tubules, as we said, we can have in testicular lobules many, 1 to 4 tubules, and they are convoluted tubules. So the space between those tubes is called the interstitial space, and this space, here we talk about the interstitial space, and this space between the seminiferous tubules will contain the Leydig cells, which will produce the testosterone and the androgens. So they synthesize the androgen. They have an endocrine activity, and they synthesize androgens, testosterone. Okay, and this here we have the Leydig cells. Okay, they are the Leydig cells represented in the interstitial space. This is the interstitial space, and we have also blood vessels that reach this interstitial space, and that will supply the seminiferous tubules with oxygen and nutrients, and will exchange hormones with the Leydig cells also. Okay, the testosterone produced by the Leydig cells can pass into the circulation directly by going to the blood capillaries or vessels, and we have also lymphatic vessels reaching this interstitial space. Okay, is it clear?
دكتور يعني الليديج موجودين بقلب اللوبيولز بس برات السمنفيرس tubules صح؟
Yes. So the Leydig cells, they are outside of the seminiferous tubules, between them. Okay, in the interstitial space.
ايه بس انه احنا كل شيء عم نحكيه هلا هو بقلب اللوبيولز مش هيك؟
Yes, because between the lobules you have the tunica albuginea, the septa of the tunica albuginea. So we are talking here inside of the lobules. We have the interstitial space between the tubes and containing the Leydig cells. Okay.
اوكي دكتور كلير.
يعطيك العافية دكتور.
دكتور.
Yes.
بدي بس اسألك عن الاندروجينز، شو هن؟
شو هن؟
الاندروجينز أي نوع هرمونز؟
In general, I mean the androgens. You wasn't present and for the chapter 2.
بلا يعني هن التستوستيرون وهيك.
Yes, the testosterone, the the dihydrotestosterone, the DHT, the DHEA. Okay, those are the androgens. Yes.
Okay. هن كمان من ضمنهم الاستروجين والاندروجين
No, the estrogens. لا, the progesterone is not an androgen and the estrogen is not an androgen. The androgens are only the يعني considered the male hormones يلي هن DHEA, the testosterone, the DHT and the DHT.
Okay, thank you.
Yes.
estrogen
Yes.
هن ال precursors للأستروجين.
لحظة, I didn't hear you, sorry.
ما الأندروجين هن precursor للأستروجين.
Yes, the androgens are the precursors of the estrogen, but they are two different subfamilies. يعني كلهم هن
Yes.
steroid hormones, but the androgen is a subfamily under this يعني تحت هيدا الفاميلي الكبيرة اللي هي steroid, and the estrogen belong to another family, to another subfamily. Okay. You didn't study the chapter 2.
Leydig cells.
Let me go back here.
لا دكتور بدي أسأل عن الليديك سيلز مش عن chapter 2.
I know. Okay, what's your question?
إنه الليديك سيلز هني بس responsible عن إنه بيعملوا أندروجين، بس شو هو الأكشوال سيل المعنية بالبرودكشن تبعت السبرم؟ يعني فهمتي علي كيف؟
Yes, we will see this in the chapter. The sperm cells, they are produced from the spermatogonia. يعني you have the germ cells. ما نحن حكينا بالembryogenesis, we have the primordial germ cells that
Yes.
they will differentiate to give the spermatogonia, and the spermatogonia will differentiate to give the sperm cells. Okay, يعني ما خصهم بالليديك cells. هن الجيرم cell line والليديك cells is a somatic cell. The leydic cell is a somatic cell. يعني they are مش الليديك cells هن يلي بيعطونا the sperm cells, but the production of the androgens by the leydic cells will regulate or will contribute to the regulation of the production of those sperm cells. Okay.
Okay. Yes, doctor. وضحت.
Yes, we will see this late in the chapter. Okay. هلا, are you seeing whole slides on the hormones?
Yes.
Yes.
Yes. I go back to this slide here. Here you have the hormones, the steroids. يعني under the family of the steroids, you have the progestogens, like the progesterone. You have the androgens and the estrogens. The androgens, they are a different family from the estrogens. Even though the estrogens are coming or have the androgens as precursors, but the estrogens are considered as a different family. Okay, and the androgens هن the DHEA, the androstenedione, the testosterone, and the DHT. Well, estrogens, they are the estrone and the estradiol. Okay.
Yes, okay.
Okay. I go back to the chapter 4. This is a cross section of the seminiferous tubule, and this is only for you to see the basal lamina here and the lamina propria, where we have like where you see the arrows, they are the peritubular or the myoid cells here, like where they are showing you them with the arrows. Okay, so those are the testicular seminiferous tubules, and this is the interstitial space, and this is the basal lamina and the lamina propria of seminiferous tubule. Now we will talk about the stages of spermatogenesis. When we talk about the spermatogenesis, this is the process by which the spermatozoa are produced from stem cells. Okay, so the process through which we have the production of spermatozoa from stem cells, which are the spermatogonia, is called the spermatogenesis. Okay, and this process have a duration of 74 days, almost 74 days. Okay, the entire process from a spermatogonia, from an immature, from stem cell to a spermatozoa, the process takes 74 days. And we have three main stages during the spermatogenesis. The first stage is the mitotic proliferation. During this mitotic proliferation, we will have the production of the spermatogonia, and the increase in number of the spermatogonia. Okay, they will increase in number, and so they divide and they are produced. Then we have the meiotic division, which will produce the spermatocytes, because as you remember, we saw it in the chapter 1, the spermatogonia will undergo, when they enter in meiosis, the spermatogonia will give the primary spermatocytes, which will enter the meiosis to give the spermatids. Okay, and then the last step is the spermiogenesis. The spermiogenesis is the differentiation of the spermatids into spermatozoa. Those the spermatids will differentiate to give mature spermatozoa. Okay, this is called the cytodifferentiation process of the spermatids. Is called the spermiogenesis. Now we will start by talking about the mitotic proliferation, and how the spermatogonia are produced, and then how they will divide. So as we said in the chapter 3, we have primordial germ cells in the embryo during embryogenesis that will reach the genital ridges and will proliferate inside of those ridges. And once they reach the genital ridges and they will proliferate inside of them, we call them gonia. Okay, those PGC, we can start to call them gonia, and later they will be termed prospermatogonia. Okay, you don't have to know those details also, but it is to clarify the ideas. So here inside during embryogenesis, the PGC will become prospermatogonia, or precursors of spermatogonial cells, of what will be the spermatogonia. Now the embryo will having prospermatogonial stem cells. Okay, at birth it will have prospermatogonial stem cells, which are quiescent, because those spermatogonial cells or stem cells before birth, they divided a little bit for a while, and then they will stop dividing. They will be quiescent, and the newborn will get born having those prospermatogonial cells, which are quiescent and not dividing, and they will remain quiescent until few days after birth. Okay. So those spermatogonia, they will proliferate for a while, and then they will be quiescent, and they will remain quiescent until few days after birth. Now after birth, those spermatogonia, or prospermatogonia, excuse me, they were in the middle of the seminiferous tubules. If you remember, we talked about cords, seminiferous cords. Okay, so the cords are not hollow from the inside. During all the embryogenesis, those seminiferous tubules will remain filled, and there is not they don't contain a lumen during the embryogenesis, and it's only after puberty that they will become hollow and they will have a lumen. Okay, so after birth, the cells, the prospermatogonial cells will move. They will migrate from the center of those tubules. They are not hollow yet. Okay, and they will move to localize themselves themselves adjacent to the basal lamina. They will sit close to the basal lamina, okay, and then they will divide a little bit to give the spermatogonia, okay. So those prospermatogonial stem cells will divide and differentiate a little bit to give those spermatogonial stem cells or the spermatogonia, okay. And then those spermatogonia, they remain quiescent until puberty. So the formed spermatogonia will be quiescent after birth until the puberty. Quiescent يعني they don't divide, okay, not dividing. So they will remain quiescent until puberty, and until it's only at puberty and starting puberty that they are activated and they enter the mitosis, so they can start to divide and later to differentiate to give the germ cells, okay. And they are activated, they enter meiosis in the basal compartment because they are localized near to the basal compartment and near to the basement membrane of the seminiferous tubules, okay. So they enter meiosis after puberty. They enter a mitosis after puberty, and they start to divide and then to differentiate to give the germ cells. So regarding the spermatogonia, they are generated a little bit after birth, but they remain quiescent, not dividing after birth. They only divide, they start to divide after puberty. Those spermatogonia, in the first chapter, we talked about spermatogonia and spermatocyte, the primary and secondary spermatocytes and spermatids. But in fact, the spermatogonia, there are more than one type of spermatogonia. We have the type A and the type B spermatogonia. And under the type A, we have also two other types of type A spermatogonia. We have the type A dark, the dark type A, which we write as AD, D for dark, okay, and the type pale, pale type A spermatogonia. Pale, we write it as PAP. So we have the AD spermatogonia and the AP spermatogonia. The pale type A spermatogonia and the dark type A spermatogonia, and they are called they are called dark and pale according to the state of their chromatin. It was observed and how it was observed under the microscope. So the chromatin of the type AD spermatogonia is very dense and granular. Okay, and those AD cells under the microscope, their nucleus will look very dark. And the type AP spermatogonia, they have their chromatin is more diffuse inside the nucleus. They have a less condensed chromatin than the AD type, and their chromatin under the microscope will look more diffuse inside of the nucleus, and this will give a paler color for their nucleus than the AD type. Okay, so that's why they call them AD and AP. Okay, so we have the type AD, the type AP, and the type B spermatogonia. The type B spermatogonia, they are more differentiated than the type A spermatogonia. And as we said, the spermatogonia are located at the periphery of the seminiferous tubule.